Welcome to the Cancer Research Institute Cancer Immunotherapy and You patient webinar series. Today is Thursday, February 8th, 2018, and the title of today's webinar is Checkpoint Blockade, Taking the Brakes Off the Immune System. Before we begin, I'd like to thank our generous sponsors whose support makes this webinar series possible: Bristol-Myers Squibb, and additional support from Regeneron, Sanofi Genzyme, and Adaptimmune. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute, a nonprofit organization founded 65 years ago with the mission to save more lives by advancing research that aims to harness the immune system's power to fight all types of cancer. This work has contributed to the development of life-saving immunotherapies for a variety of cancer types. We present this webinar series to help patients and caregivers who are interested in learning about immunotherapy understand what it is and how it's different from other types of cancer treatment, to provide you information on the latest developments in research and treatment, and to connect you to cancer immunotherapy clinical trials. Now it's my pleasure to introduce today's expert speaker. Dr. James Allison is a true rock star of cancer research. He is the Vivian L. Smith Distinguished Chair in Immunology, Executive Director of the Immunotherapy Platform, and Director of the Parker Institute for Cancer Immunotherapy at the University of Texas MD Anderson Cancer Center in Houston. Since 2011, he has served as Director of the Cancer Research Institute Scientific Advisory Council. Dr. Allison is an internationally recognized scientist whose pioneering work in immunology and tumor immunology has garnered numerous awards and prizes, including the 2018 Jesse Stevenson Kovalenko Medal from the National Academy of Sciences, the 2017 Balzan Prize for Immunological Approaches in Cancer Therapy, the 2015 Lasker-DeBakey Clinical Medical Research Award, the 2013 Lloyd J. Old Award from the Cancer Research Institute and American Association for Cancer Research, and the 2005 William B. Coley Award from the Cancer Research Institute, among many others, and they just seem to keep coming. He's best known for his groundbreaking discovery in 1982 of a molecule found on our immune system's T cells called a T cell receptor, which allows these important defenders of the immune system to identify their targets for elimination. The National Academy of Sciences described this discovery as one of the three most important findings in immunology over the last 20 years. In the late 1980s, Dr. Allison discovered another key mechanism of T cell activation that requires the presence of a second molecular signal in addition to the target antigen, a process called co-stimulation. In a watershed moment in cancer immunology, Dr. Allison and his team of scientists went on to identify a cellular pathway called CTLA4 that is responsible for shutting down activated T cells. This checkpoint process protects us from potentially fatal autoimmune disorders that can result from an overactive immune system, but in the treatment of cancer, this checkpoint can block T cells from attacking tumors. In a seminal paper published in 1996 in the prestigious journal Science, Dr. Allison and others in his lab showed that blocking this checkpoint with an antibody enhanced the immune response to cancer, demonstrating in mouse studies that this approach could result in complete elimination of tumors. This work eventually led to the development of ipilimumab, which the FDA approved in 2011 for the treatment of patients with metastatic melanoma. It was the first checkpoint to receive FDA approval, paving the way for approval of other checkpoint inhibitors for a variety of tumor types. And so with that, Dr. Allison, thank you so much for joining us. It's truly an honor to have you here with us, and we're so glad that you could be with us today.
Well, thank you, Brian, for that nice introduction. It's a pleasure to be here and share some of the excitement in this field that's going on right now.
So let's start at the very beginning. What got you interested in immunology in the first place, in science?
Well, I was a graduate student in biochemistry starting in the late 60s, early 70s, and I took an immunology course. It was taught by this guy who was an antibody guy. Because at the time, antibodies were pretty well worked out, but there were these curiosities called T cells that had just been discovered. And my professor taught the rudimentary things about them, which not much was known, except they were cells that went around the body looking for things that oughtn't to be there and eliminated them. But how they worked, there was nothing known about that at all. And I was just fascinated by by that process, how you could have cells going around, checking out other cells in the body, recognizing something that ought to be there and deal with it and not hurt your normal cells. And so I, at that time, after that, got my Ph.D. again in biochemistry and decided I was going to switch to immunology, which was delayed for a while. I did a postdoctoral work working in an immunology lab, but I was still doing protein biochemistry and purifying stuff. And anyway, when I got my first faculty job, I again, it was at MD Anderson, but at a lab, not in Houston, at a lab near Austin. Anyway, I just decided from now on, I'm working on T cells. I'm going to figure these little guys out, try to unlock their secrets. That was in 1976 or so, and I've been doing
now
it.
I've of course, you and I have known each other for some time through our relationship in the Cancer Research Institute, and I'm very familiar with your story that's been widely reported in news outlets about your in the very beginning, you weren't even thinking about this as a cancer problem. You were really just fascinated with the basic immunology. So at what point did you realize that your work was going to have an impact or could potentially have an impact in cancer?
Yeah, that's a very interesting question, and I've thought about it a lot. I guess the thing was when I was about 10 years old, my mom died of a lymphoma, and I was holding her hand when she died, and it made quite an impression on me, as you might imagine. And then within a few years, both of her brothers died of cancer, one of melanoma, the other of lung cancer. And I just, everybody I think has had friends and relatives, and I just wanted to really do something about it, but I didn't really. I mean, very little was known about cancer then either. I mean, even less than about T cells really, except that it could be caused by chemicals or whatever. But I just decided. I mean, the idea that the immune system could fight cancer had been out there for a long time, since William Coley, as you know, and in the '60s began to be real efforts to actually use it to treat cancer. The first person to actually proposed that the immune system might have a silver bullet, that could be used to treat cancer was Paul Ehrlich in 1906. I got the Ehrlich Prize from the German Immunological Society several years ago, and they showed me his lab, which is still there. It was open to a page in German, but he'd drawn an antibody molecule, which very crudely, and then just wrote there, and had a cancer cell. And it turns out, all these years later, he was right, except that the ones that work best are, as we will discuss, aren't actually the antibodies to the cancer cell itself, but are to the immune system. But I always, having decided to do that, I, keep in the back of my head that the immune system can attack cancer, just decided in order to make it work right, in order to manipulate it in the way we needed to, I needed to understand. We needed to understand that the details of how it gets turned on, how T cells get turned on, what regulates them, how they get turned off, and only when we know the details of that are we going to be able to do anything to cancer. So various times during my career, we've found things that I thought might lead the way, and I'd jump into real cancer immunology for a little while and see if they worked. But the first couple of times didn't work so well, and so went back to the basic science. And finally with CTLA-4, we began to work out how it turned T cells off, and just exactly how that worked. And what I try to do in translational work is concentrate on the basic science until I really feel that I know it, and then take a little time off, and then try to think about how can we use what we discovered here in the real world to treat cancer. And so that's what I did. Was after we found CTLA-4, put my feet up and really thought, putting pieces together, A few years before, we'd shown that tumor cells, in part, are invisible to the immune system because they can't provide that. They have antigens, so they can activate the antigen receptor, which is like the ignition switch of the immune system, but they don't have the molecule that pushes, that gives the co-stimulatory signal. We showed that by putting that molecule into tumor cells and found they got rejected immediately. So they had antigens, they were just invisible. So we tried to treat cancer by putting B7 into tumor cells and then immunizing mice. That actually worked. You could, the tumor cells, if you put B7 in and put them in a mouse, they'd get rejected. You could give the mice then tumor without any modification whatsoever, and they'd be rejected as well. The problem is that it was specific for that tumor, and the second problem was the tumor cells had to be alive. We tried to inactivate them a number of ways, and it just destroyed their ability to co-stimulate. So I said, well, okay, nice try.
I'd like to go back and talk about, you just mentioned gas and brakes. The title of the webinar is Taking the Brakes Off the Immune System. Can you just say something about kind of what was the prevailing approach to immunotherapy in cancer? Why wasn't that working, and how did checkpoint blockade change that?
Yeah. Well, the earliest attempts to use the immune system involved cytokines and IL-2 and stuff like that, just to try to generally activate the immune system. There were some responses, but that was so toxic that it didn't really prove to be practical. And then once tumor antigens were discovered, people began to try to vaccinate therapeutically, not like you do with polio, but more like you do with rabies. You've already got something. You want to rapidly get an immune response started. Treat something that's already there, and those just didn't pan out. I think there were a number of reasons. One of them is that at the time we didn't really realize how complicated T cell activation was. Nobody really realized that there were these a negative signal. The CTLA-4 molecule, for example, is not expressed in a T cell until it gets activated, and then you activate a T cell. What we know now is that turns on this go pathway that tells the T cell you start dividing as much as you can, because you've got to go from a few dozen to hundreds of thousands of T cells with a certain specificity within a week or less. So that's really a lot of cell division. The cells are just cranking along. But also what we found in 93, 94 was that this molecule CTLA-4 is turned on, and it actually serves as the brakes with time. It stops the immune system. At the time, people thought that activated T cells just died off. That was largely through a process called activation-induced cell death. This checkpoint was the first thing, and one of the things that occurred to me is that after the first dose or so of a vaccine, it was likely that what was happening is it was actually giving a signal that said stop. It's probably activating the CTLA-4 pathway more than the GO pathway, and it might have just been counterproductive in ways that it was being done. And because it was a lot of doses of vaccine really quick, I think that they may have just been turning it off. And so, I had the idea, putting together the fact that tumor cells can't co-stimulate. I said, okay, well, the tumor gets a head start because it's invisible until some tumor cells start dying. Maybe the tumor gets so big it can't get enough oxygen or can't get enough glucose or whatever. That causes inflammation. The immune system comes in, the innate immune system, to clean up the debris. And then dendritic cells pick up molecules, present them from the dying tumor cells, present them to naive T cells, along with the co-stimulatory signal. So that starts that off. But as I said, then CTLA-4 comes up. And so it shuts them off. And it's sort of like a clock, And so my idea was, it's a race, and the tumor had a head start. And so, the CTLA-4 is there to protect you against your immune system, because you have to stop that process of rapid division, or it'll kill you. Later, me and others showed that was the case. If you get rid of the CTLA-4 molecule,
That's just
mice, the immune system kill them
no
because they can't
break,
stop
right? You just
dividing
your
these
brakes are
strong
cut
T
and that's
cells.
it.
So, from there it was a relatively easy step. Yeah, yeah, your brakes are going to come on. You just got to suspend them temporarily. You don't want to suspend all of them all the time, or it's catastrophic. But you just do it temporarily, and sure enough, that worked. I said, okay, well, let's try this in mice. And so we made an antibody and gave mice tumors and injected this antibody, and the tumors just melted away most of the time. Some of them. Others would have to add radiation or chemotherapy or vaccine properly. And anyway, and that was a compelling notion to me for several reasons. One is that in general, one of the things that the immune system offers is memory and adaptability because it's a living thing. So if the tumor changes, it can change. But specifically, what this offered was, you're temporarily suspending the brakes, but you're you're not treating the cancer cell. You're not trying to kill the cancer cell. You're trying to release the immune system so that it can go out and eliminate the cancer cells. It's not so much, people use the word harness a lot. It's not harnessing so much, it's just unleashing. At least this particular approach is. So that being the case, it should work against all cancers because the immune system doesn't know whether cancer cells of breast cancer or kidney cancer. It doesn't know whether it's caused by a RAS mutation or BRAF mutation or whatever. It just knows that it has things on it that shouldn't be there. And so, that was pretty compelling. A universal cancer treatment. Wow, too good to be true. Turns out it is for reasons we can talk about later because some cancers just don't respond even though they really ought to. But the second thing is that whole process is started again by tumor cell death alerting the immune system that something's going on. And other therapies, chemotherapy, radiation, the new targeted growth factor inhibitors, pathway inhibitors, they all work by killing tumor cells. So it also occurred to me that what we could do, if you take one of like let's say a molecule that specifically targets this mutation in the molecule called BRAF that's found in more than half of melanoma patients. An inhibitor of that molecule will shrink the tumors in about 90% of cancer patients. But of course, it grows back because the tumor adapts to it and escapes it. But if you added CTLA-4 when the tumor cells are dying, you can get the immune system to detect all the other mutations and things that, in addition to the BRAF molecule, oughtn't to be there. So you make one molecule, one target into dozens perhaps. And the second thing is you can give that molecule memory in the form of the T cells that are left over. And so one of the things that occurred to me early on was this could be a type of therapy which could be combined with almost any other cancer therapy to really make them work better.
So you mentioned, the tumors melted away in the mice. And, there have been other examples in the past of treatments proving, effective in mice, but doesn't translate so well into humans. What made you think that your treatment was different or that there was a greater chance of success in humans?
Yeah. Well, of course, I wasn't sure, but I wanted to make sure that we weren't just doing a peculiarity thing. A singularity is what we call it. Something works in one configuration, never works again. So we did it against many, many, many different kinds of tumors and many different strains of mice. And like as I mentioned earlier, it didn't always work. It tended to work against chemically induced tumors just by itself, just a single dose of the antibody. With other things, I said you'd have to combine it with something else. But it just, it didn't work partially. It didn't slow tumors down most of the time. You just cured them. Perfect experiment. The mice are feet up, instead of walking around. They're walking around in the ones that you treated. So you don't need statistics or anything. It's generally 90 to 100%, or it's nothing. So I really wasn't sure, but it was powerful enough, and I was excited enough that I just began to try to get somebody to help us move it into humans. But it was hard because, as people did say all the time, they said, well, anybody can cure cancer in mice, but you don't know if it's going to work in humans or not. And many people were skeptical, and so it took about 3 years before we finally succeeded in coming up with a company ready to take a chance and go to the expense of
That's a real testament to your persistence and your
start the
belief
first clinical trials.
that you had found something meaningful. And you know, sometimes the best ideas are often rejected in the very beginning. And now, of course, look at the field of cancer immunotherapy, and it's all thanks to your work, discovery of this checkpoint blockade. Let's talk about the patient experience of receiving checkpoint blockade. How is it administered to patients? What does it look like? How's it administered? How does that treatment compare to other types of treatment like chemotherapy?
Well, and checkpoint blockade is an outpatient treatment. Patients go in different intervals depending on the drug with anti-CTLA-4 or ipilimumab. It's named by the FDA, unfortunately. People just go and sit in a chair for about an hour, and it's infused into a vein, and then you remove the shunt, if you will, and people go home. There are adverse events sometimes, but that's about it. The adverse events are something important and worth talking about a little bit. With chemotherapies, typically, I think everybody's aware that people treated with chemotherapies, lose their hair. They have a lot of trouble because the cells that normally line the gut are sloughed off, they're killed. Almost any rapidly dividing cell is destroyed because that's how chemotherapies typically work, is they kill rapidly dividing cells. And they happen to kill tumor cells a little bit better than normal cells for that reason. But basically everybody that gets chemotherapy has these stereotypical toxicities. Where with the checkpoint blockade, there are toxicities which we didn't expect, actually, because we never saw anything in mice. But they're typically, not to trivialize them because they can be quite dangerous if untreated, but they're things like colitis, very bad diarrhea, pneumonitis, trouble, inflammation of the lungs, a lot of itises, hepatitis, temporary things. These are all inflammatory reactions. People say they're autoimmune. They're really not, because you can treat them with steroids and they go away, and then you wean the patient off the steroids and they don't come back. Having said that, there are a very small fraction of patients that do develop serious autoimmune situations, such as we know now that tens of thousands of people are being treated. Very rarely people can develop diabetes, for example, or some other bad things. But that's how it differs. Like something like 40% of patients that are being treated with single agents really don't develop much of any toxicities at all. Others can have rashes or whatever. So
thanks for touching on side effects. That was going to be my next question. It's often a question that patients want to know when considering treatment with an immunotherapy. What were some of the benefits you saw, and kind of how many patients seem to respond to treatment with checkpoint blockade?
Well, the first trial was a phase 1 trial, and it was in, I think, 14 patients with melanoma. It's what's called a phase 1 trial. The way that trials are typically done is phase 1 is for safety. You're not looking for any kind of clinical signal. Phase 2 is trying to find the dose at which you begin to see some effect on the tumors. And phase 3 is compare it to whatever the standard of care is. Well, in the phase 1 trial, the very first time it went into people, there were, I believe, 3 out of 14 patients had responses in melanoma. One of whom, actually, I met, and in 2001, she was being treated for melanoma. At the time, the median survival after diagnosis of late-stage melanoma was 11 months. So it was pretty much a guaranteed death sentence. There were no drugs that had been ever shown in any kind of proper clinical trial to improve survival. But anyway, to get three responses in a phase 1 was just unheard of at the time. And one of those patients is still alive now. 17 years later, it was 2001. She was, and I've met her and chatted with her briefly. She agreed to take it because she'd failed everything else, and her doctor, UCLA, got him Tony Rivas, who's a good friend and outstanding oncologist. Said he had this drug. You want to know if she'd try it. Had never been to people before. It had the potential to be dangerous. And she said, I just want to live a couple of months. I just want to see my son graduate from high school. Anyway, she's still alive almost 18 years later. So she's gotten a chance to see him graduate from high school, college, start his own family.
I imagine, I mean, you're, for the people watching, you're not actually a doctor. You're not there often interacting directly with the patients. You're a lab guy. So what was that experience like for you, seeing, people who are alive because of a discovery that you made?
Well, it was, it took a while, actually. I mean, I saw the numbers, that there were a lot of trials going on. Of course, I was consulting, advising this company Medirex, and then eventually Bristol-Myers Squibb. But I moved from California to New York to work at Sloan Kettering because I wanted to be around where the trials were being done. But I didn't meet a patient until about 2004. It was the first patient I actually met. Until then, it was numbers, But I saw the numbers and they impressed me, because I thought, well, that could have been my mom, and it could have been my uncle. I recently lost my older brother, to metastatic prostate cancer. And, I thought, well, it might be, but it was just numbers and I was inferring it. But I think it was 2005. While I was at Sloan Kettering, I got a call from Jed Walchuk, who is an oncologist who was there, well associated with the CRI. He called me, said, come down to the outpatient clinic. I said, why? I'm busy. And he said, just come down here. Don't ask questions, just come down here. It's about 4 blocks away. And so I said, okay, walked down there and asked where he was. They told me. I walked into this examination room, and he was there with this woman, her husband, and her parents. And she came over and grabbed me and hugged me. Everybody started crying. I started crying, and she was in her early 20s, had just finished college and got married. Was just starting her life, and that she'd failed everything as well. She had massively metastatic cancer in her lungs and liver and brain, and she'd been treated again in this trial that Jed was doing. And a year earlier, all her tumors had gone away, and so she was in for her first checkup, and just been told that she was still tumor free, and naturally everybody's really happy. So she was pretty emotional about it. And Jed said, "Well, the guy that invented this is here. You want to meet him?" And so that was why he called me, and it's really a nice thing to do because I've learned a lot.
And one of my cycling buddies, she and I and her husband Rob like to cycle together whenever we can. So
and,
it's amazing.
So. Yeah, she's. But as I say, I learned a lot from her first. I happened. I don't know how many years later. I keep up with her. She sent me. Occasionally, she sends me photos. She has 2 children now, as you know, and now and then sends me photos. And at first, though, she told me 2 things from during that early period after her treatment. One of the reasons she had been so emotional when she met me was that she was positive that she was going to be told that her cancer had recurred. She'd been through such an ordeal before. She was assured they were going to say, no, it came back. And then every year after that, she went through the same thing. Just being afraid. And also the doctor said, well, you probably shouldn't have any children. But, after a while, she just said, well, to hell with this. I'm going to get on with my life. And, I'm not going to worry every day looking over my shoulder. And that meant a lot to me. A few months after she told me that for the first time, I was at a meeting and people were started talking about how, well, this immunotherapy has turned cancer into a chronic disease that's manageable. That's not something that is greatly reassuring to somebody who's suffered from what was essentially a death sentence back when this work started. And I remember that I was at a meeting actually it was a few years after that, but a study came out which was a follow-up of almost 5,000 patients for whom there was 10 years data. And it turned out that 22% of melanoma patients that received a single round of treatment of ipilimumab were alive a decade after their initial treatment and after it had stopped. And so I talked about that in one of my lectures and said, for all intents and purposes, these people are cured. And I got admonished by a surgeon who said, well, Dr. Allison, you can't really say that. You got to be careful because, cured means there's no cancer anywhere in anybody's body. Well, that's kind of a high bar because you don't know. There may be one cancer cell there somewhere, but if somebody's been alive for 10 years with no more therapy, it's time for them to quit worrying about it, in my opinion. And that's what Sharon did after a while. She just said, I'm cured. I'm not going to. And so anyway, I said to this doctor, I said, well, one thing is, you're talking about a shadow on a CAT scan. And there may be things smaller that you can't see on a CAT scan. That's true. But that's a shadow on a CAT scan. And while they're not regularly biopsied, in many, many cases where they have been biopsied, they're what are called granulomas. It's scar tissue left over from where the battle between the immune system and the tumor cells went on. They're not cancer. They're, ruins of war, if you want to look at it that way. So I told this guy, I said, well, you know what, though? That's not the way that the patients look at it. And remember, you're treating a patient. You're not treating a CAT scan. And it's important that, nobody dies after about 3 years on these therapies. If you make it 3 years, you're pretty much good to go. That's what these studies have shown for at least a decade. And there's, as I said, this one woman from the phase 1, also named Sharon, incidentally, is alive. She was treated in 2001.
You mentioned that about the number was around 22% of people having still living after all that time. Why doesn't what do we know about why it doesn't work in some patients or a majority?
Yeah, well, that was one of the puzzles because in the mice, it worked. If it worked, it worked in all the mice. But they're genetically identical, have been raised together. All this, so they're not very many variables, but people are all different. But there's, initially I thought, well, maybe it's because the drug only works while T cells are being primed. After there's been some tumor cell death, and the T cells are getting primed. That's when the CTLA-4 gets turned on. So you've got to act during that process. And if it's not happening when the drug's on board, then it's not going to work. It's a theoretical possibility. It may be part of it. There are others, but of course, one of them, which is the more interesting one, is that maybe there are other checkpoints because CTLA-4, when we found it, Jeff Bluestone's lab and mine actually in 1993-94 was the first and only known checkpoint. But after the first clinical data started coming in 2001 or so, well, even before that data started coming in, there, of course, with the stories of success, not just in melanoma actually, but many other indications as well. There were small trials that showed it also worked in bladder and kidney cancer, some responses in lung cancer, some responses in prostate cancer. So people began looking for other checkpoints. And it turned out there was this molecule called PD-1 that had been identified in the early 90s. It had a really obscure function. Program death 1 is what that stands for. But its function was really not very well worked out until Gordon Freeman and Arlene Sharp at the Dana-Farber cloned two molecules, which were the things that bound to PD-1 and showed that it was another checkpoint. And that, of course, led to another researcher, Li Ping Chen, has shown that one of those receptors could be expressed on tumor cells, and then it inhibited T cells. So all that biology came together, and so there was a mad rush, to get PD-1 in the clinic. And sure enough, it also works very well in about 28% or something of metastatic melanoma patients. And so that, of course, was cool. Now we know it too. Well, I won't go into the mechanisms of how it works, but it's very different than the way CTLA-4 works. So we reasoned that maybe if you put them together, it would be at least additive. So we did in mouse models, and sure enough, they were additive. So we could, in situations where neither one of them worked very well by themselves, together they were much better. So that led to combination trials in humans. And again, early trials showed that the response rate from PD-1 was about 28% or so. But when you combined it with anti-CTLA-4, the response rate was over 50%. The encouraging thing now is that the 2-year survival, and I believe it's also the 3-year survival, is a little bit over 60%. And so if the rules, if the ipilimumab effect is dominant,
that would be wonderful. Of course, we're all
when
hoping for 100%, but
follow-up for those patients, it very well may be that 60% now of melanoma patients, Well, that's the challenge now. I mean, to me, that's the challenge. Is we need to get it up to 100%. There's a famous, well, it's a graph that I think that Pam Sharma and I started drawing for the first time. Usually you draw a survival curve, and the whole goal of a trial is to see if the median survival, where you plot percent alive versus time. If you treat patients and the median survival is longer, is of 50% of the patients, if that's significantly, which is usually measured in weeks or months, longer than the standard of care or placebo or whatever. What ipilimumab showed is it did that. It improved the median survival 4 months, but then the curve started flattening out at that 20% number in about 3 years and stayed there for 10 years, which is the longest, well, they're still being followed up. It's at least 10 years. That's a minimum, because people after about 3 years aren't dying from melanoma. So that's called the tail of the curve. And so that's the goal that immunotherapy has brought to cancer. We don't look so much for an increase in the median anymore. We look for the percentage of the tail. And that whole, like you say, the goal is to get that as close to 100% as we can. And also, there's many tumors in which the response rate is lower, or even so low you can't.
You mentioned in the early mouse studies that chemically induced tumors seem to respond better to this sort of treatment. What do we know about cancers in humans? Are there some that are more likely to respond to this type of treatment than others, and why is that?
Yeah, you're correct. And so we published several papers on this, and basically whatever, sometimes we'd use cryoablation, radiation, whatever, and get the response rate up from the monotherapies. But usually the cancers where we had to add a second agent were what we call spontaneous tumors that arose in mice for no really known reason, but the experimental tumors that most people work with were induced by carcinogens. So at the time, I thought, well, maybe they have more antigens because that was what everybody thought. But now we know that the tumors like melanoma, lung cancer, head and neck cancer, bladder cancer, those are all caused by mutations. The ones of the lung, head and neck, bladder, etc., from contaminants, from carcinogens in cigarette smoke, melanoma from ultraviolet radiation-induced mutations. And those tumors were historically very difficult to treat in humans because I said, well, when cancer biology finally showed us that it's mutations that cause cancer, it was realized that those kinds of cancer had lots and lots of mutations because of what caused them. So what happens is you start getting mutations, and most of them probably don't matter, but when you finally have a mutation on a molecule that's involved in normally in controlling growth, and you knock out a control point and get a signal that tells the tumor to grow, a cell to become a tumor, just keep growing. It's called a driver. And anyway, those are the ones that you can treat sometimes temporarily, but they're so heterogeneous, they change, and you can't really deal with them. So, but that same feature, those ones with the high mutational load, that made them so difficult to treat by conventional therapies, makes them sitting ducks for the immune system because they have so many mutations. They result in the formation of what are called neoantigens that we know now the immune system tends to be preoccupied with cancer. So that's exactly what it's hitting. Whereas you get other tumors, such as a lot of leukemias or glioblastoma, they have low mutational loads or somewhat colder. So they're more of a challenge. So that's the challenge, is to deal with cold tumors that are cold either because they don't have mutations. You only need one good one, it seems, but the more you have, the more chances are you have getting a really good one, according to the work of Bob Schreiber. The other feature is whether T cells come in to a tumor. The colder ones that have fewer mutations, tend to not have as much inflammation from the immune system as others. Prostate cancer, on the other hand, has a relatively small number of mutations, and normally doesn't have many T cells in them, but you can drive T cells into them with ipilimumab. And what we found is that upregulates the ligand for PD-1, so that protects them. But if you use the combination, it should be much better. So
and my understanding, correct me if I'm wrong, is that these signals, such as the level of infiltration by T cells that are already in the tumor, or the number of mutations, or the presence of things like PD-1 or PD-L1, that these are signals that oncologists are beginning to use more and more to help pre-select and pre-screen patients to predict the likelihood of their response to these treatments. These are called, I think they're called biomarkers. This is a word that's kind of coming up more and more, and I know it's an area of intense study right now to try to find these truly predictive biomarkers, because that influences your treatment decisions then, of course. Are there other biomarkers that we're looking at today?
Right. Well, yeah, as you say, the biomarkers, well, there can be biomarkers of toxicity as well. But the ideal biomarker, as you say, is something that you can look at in a patient that hasn't been treated and say whether the patient will respond or not. Unfortunately, we're not there yet. There's things that are correlated, like a high mutational load. On the other hand, there are patients with low mutational loads that respond quite well. High PD-L1 levels can do it, but there are a lot of patients that don't have PD-L1 in biopsies, at least, that do respond. And so there's a balance in using those biomarkers. If you use them rigorously and trying to decide who to treat, at some point you're going to start not treating people who might have responded. And if there's no alternative therapy, that's not a very good thing. You have to decide where you're going to do it. PD-L1, for example, I believe, if you have it, the chance is about 40% of responding to PD-1 in melanoma. If you don't have it, your chances are about 15%. Where do you want to decide on who to treat just on that? It can be argued both ways, but bottom line is, there just isn't anything, in my opinion, that we have right now that gives you sufficient predictability to make that decision. However, there's another kind of biomarker where you look after you initiate treatment and look for collection, not a single thing probably, but a collection of changes, like more CD8 T cells in a tumor, more PD-L1 or whatever, and see if that happened. And if there's been studies by Jen Wargo, for example, here at the Anderson, that shows that there is a signal associated with on-treatment specimens. It's pretty good at predicting who's going to go on and treat or who's not going to respond, and you should switch treatments immediately. Another marker like that is a molecule called ICOS that Pam Sharma found many years ago, and what Jed Wolchak and when I was at Sloan Kettering, what we found in a retrospective study of melanoma patients is that patients that didn't show an increase of that molecule, even in the blood, this is one thing that happens in the blood. Most don't, but it's one thing that happens in the blood. Also happens in the tumor. But if there was a rise in the frequency of these CD4 cells expressing this molecule, the patients had a much higher chance of responding. They didn't always, but they had a much higher chance. But if that didn't go up, they just really didn't have much of a chance at all. And so that's another marker that can tell you, well, if you don't see this, then you better do something else quickly. But we're still learning. What the ICOS molecule is called is a pharmacodynamic marker. What that means is that's a biological consequence of your drug hitting its target. It can tell you that something's going on, at least. Yeah. If it doesn't happen, your drug just didn't work.
So we're almost out of time right now.
Too, although again,
Dr. Allison was just wondering, the field has really come a long way in just the past 10 years in terms of clinical successes and regulatory approvals. And checkpoints, of course, are at the center stage of this kind of revolution. In addition to now, other types of therapies like CAR T-cell therapy. What do you see as, in the next 5 to 10 years, where do we need to get to, and how are we going to get there, and what's cancer treatment going to look like at the end of the next decade?
Well, where we are now, where we were 5 years ago, arguably, there were 3 pillars of treatment for cancer. Surgery, radiation being the oldest, more recent was chemotherapy. And now, finally, after a lot of debate, immunotherapy has undoubtedly met the criteria, being the 4th pillar. Will it ever replace all that other stuff? No, unfortunately, I don't think it will. Perhaps combinations of immunotherapies in some cancers, melanoma, as we talked about, is going to be pretty successful at. But other tumors, for the reasons we've discussed, are not necessarily going to respond so well. But there are other things that can be done that really aren't being tried all that hard yet. The rival field recently has been targeted therapies where you sequence the genome of a patient's tumor, compare it with their normal cells, and identify the mutations, and then find ones in certain molecules that we know can cause cancer. And in many cases, there are these all turn out to be enzymes. In many cases, there are inhibitors available for those targets. And we know, as I mentioned earlier, because of the heterogeneity of the tumors, they're not going to be curative. But if you use them to kill, a lot of tumor cells, not all, and then add the checkpoints on them, you can give them the features of immunotherapy. And I think that's what I think is going to happen, and I'm really, really urging this to happen more, is to use all four pillars in combination. That's when we're going to really have effects. One of the things that we showed years ago in a collaboration with Sara Maria and Silvio Fermenti now at Cornell was that in a model of mammary cancer that the local cancers could be absolutely cured by radiation, but they didn't improve lifespan at all because they had already metastasized. The tumors had already spread elsewhere in the body. But if you gave them anti-CTLA-4 right after the radiation, you cured almost all the mice. And so now what that group is thinking and working towards, as are a number of people in radiation therapy, is giving up with the notion that you have to kill every last tumor cell with your radiation. You say we shouldn't even be thinking of that as a goal. Let's just kill enough and do it in a way where we don't destroy the immune cells and use it, to get us to a state where immunotherapy will work. And so cancers in which immunotherapy doesn't work by itself, I'm confident that and there's already a lot of work being done in lung cancer that shows you can irradiate a single metastasis followed by, anti-CTLA-4, for example. Some studies have been done with PD-1, but the whole body responds. And I think that's something that's going to be incredibly powerful. And when we simply in a rational way work out the mechanisms of action of these genomically targeted medicines, I think, and try to find out how to make them play nice with the immune system, I think we could affect a lot of cures that we're not seeing now. So anyway, I guess that's a long-winded way of saying that I think that immunotherapy is going to become part almost
complete difference in terms of perception of immunotherapy's viability as a treatment. So I share your optimism. I know in talking with patients at our immunotherapy patient summits or on our website that hearing about this certainly gives them a lot of hope that they or a loved one will benefit from this sort of work. So that is all the time we have, Dr. Allison. So I want to thank you again for joining us today and for really walking us down the history of what has undoubtedly become one of the most revolutionary breakthroughs in the treatment of cancer ever, perhaps. So that's a real treat.
Well, there are a lot of hurdles still there, but I'm optimistic as well, and many people are working very hard, because what's encouraging is we're not there yet. We're just at the beginning, really, but we know what the basic rules are now.
Yeah, with more research, we can get there. We absolutely believe
getting
that
back to fundamentals, understanding the science.
With that, I'd like to thank once again our sponsors, Bristol-Myers Squibb, Regeneron, Sanofi Genzyme, and Adaptimmune. If you'd like more information about immunotherapy for various types of cancer, we have an informational resource on our website, cancerresearch.org/patients. There you can get connected to our other resources, including our entire webinar series, which is available on our website, or you can go to cancerresearch.org/webinars. Thank you for watching Cancer Immunotherapy and You, and we look forward to another webinar with you next month. And Dr. Allison, thank you.